right, good afternoon, everybody. Thank you so much for joining the webinar today. I'm Mark Sadam, Vice Provost for Innovation and New Ventures here at UNH. Today, we're joined by President Jim Dean, who will provide an update of how we're preparing to welcome students back to our campuses for a COVID-19 ready fall semester. We'll then be joined by Corey Taylor and Brad Miscowski, both alumnus of UNH's College of Engineering and Physical Sciences, and they'll discuss how Lonza and Moderna are teaming up to accelerate the manufacturing of a potential coronavirus vaccine. We'll finish the program with a few details of UNH's impact on the project and our partnership with Lonza, followed by a question and answer session. Questions can be submitted through the webinar using the Q&A feature at the bottom of your screen. We'll have a few opportunities to answer your questions throughout the webinar. With that, I'll turn it over to President Dean.
Thanks, Mark, and thanks everybody for joining us today. It's really nice to have you. This is the third or fourth one of these I've done, and I'm really struck by the participation and the interest that our alumni community has in what's going on at UNH. This has, of course, been an historic challenge for the state of New Hampshire as a whole and for the university in particular. And I think it's shown the value of a strong university and also a strong university partnership between the university and the state. Of course, the first priority that we've had has been the health and safety of our students and the entire community, actually the communities in which we operate, Durham, Manchester, and Concord. And so I'm sure, as everyone knows, as the pandemic started up in the spring, in March, we closed all of our campuses, we refunded room and board fees to students, and we quickly transitioned, actually amazingly quickly, transitioned to remote learning and remote work. I sometimes joke this was sort of the world's biggest social science experiment to see if we could change from regular in-class kind of learning, teaching and learning, to remote learning. And for the most part, we did. Well, we did completely, and for the most part, it was successful. And we certainly learned a lot of lessons that we're hoping to put into place as we move forward with both the combination of remote and in-person learning. I think it's also worth mentioning that this has really become an all hands on deck situation in terms of the university supporting the state. So just a few specific figures in terms of personal protective equipment. The university provided 45,000 pairs of gloves, 11,000 masks, 4,800 respirators, and other equipment to make available to the state to help in the response to the pandemic. We also stood up at Hamel Rec Center basically a hospital, a patient overflow center, which was ready to hold 250 people. Now, fortunately, things never got that bad in terms of filling up the hospitals in New Hampshire, so we didn't have to use it, but we were really happy to work with the state on that as well. We are in the final stages of setting up a testing center here at the university that will be able to test our students, our staff, and faculty. And I'm happy to share some more details. Mark Sadam, who introduced the call, has been really the point person on that, and he can give you a lot of details if you're interested in that. Interestingly, we've also hosted the New Hampshire House of Representatives twice in June. So they've come here. It's the first time in over 100 years that they've met outside of the State House in Concord, and they had, from what I've given to understand, some pretty productive sessions, and we were really happy to have them. We're really delighted with the support that alumni have provided to the emergency fund for students, which was something that we were creating just before the virus happened. And then once the virus happened, it became much more important to provide emergency funding for students. And we're really happy with the alumni response to that. And it's made a big difference in the ability of our students to transition over the past few months. Now, this isn't just a COVID-19 thing. This is an indefinite opportunity to support our students with food and shelter and other kind of emergency needs that they have. Over the past couple of months, we've also had a series of webinars featuring UNH experts and researchers on a variety of topics, including the virus itself, but a wide range of other topics as well. So as we go forward into the fall, we really have a couple of very big goals. One is to have a campus that can continue to operate despite the virus, despite the pandemic. And we've been making really elaborate plans to do that, which include flexible blended instruction, both on campus as well as online instruction. This has been possible because we've had roughly 150 people working in a set of teams around education, around classrooms, around cleaning, around tracing and testing and so on. Many faculty, many staff, and even students have participated in these teams. So we have some fairly elaborate plans about what's going to happen in the fall. In a second, you'll have the opportunity to ask some questions about that if you're interested. But for now, I want to point out that these plans are posted on our coronavirus website, which is part of the UNH website. It's pretty easy to find, so I'm not going to read out the whole URL to you. I also want to say how grateful I am to our alumni for sharing their support and talent. In a couple of minutes, you're going to hear from Corey Taylor and Brad Miscowski. And I also want to thank Mark Sedam and Debbie Dutton for their support of this. And I think this exemplifies how UNH talent is innovating to solve global challenges like this pandemic. So with that, I'll stop. And Debbie Dutton, I'll turn it over to you to see if there's any questions that people would like to ask.
Hello, everyone. Thank you for joining us. If there's questions, if you want to ask those using the Q&A chat on your screen, that's really great. We don't have any right now, so we're eager to have Jim answer some of your questions before we turn it over to Corey and Brad.
Well, while we're waiting for any questions to appear, and so the Q&A function on the screen is the one to use, Mark, why don't you take this opportunity to just say a little bit about the lab that's being set up?
Sure, thanks. So UNH had decided pretty early on that the best chance for us to have in-class instruction was to have robust testing, to be able to access testing. And early on, there was a question of outsource versus keeping things internal. And we made a decision in May to really aggressively pursue doing the work ourselves. And so we're in the process of spinning up a testing lab. So there'll be UNH folks doing the testing. We're trying to approach a very aggressive testing protocol, meaning we're going to test as frequently as we possibly can and try to get the results turned around in about 24 hours. Right now, we are 34 days from getting operations up and running. Everything is going very well. There's still a little bit of equipment that needs to arrive, but we are on path to do that. So it's been a lot of very interesting work, and what was really rewarding, I think, from my perspective, is that we made some assumptions about how things were going to progress, when testing was going to be available, when equipment was going to be available, what the FDA was going to say was okay in terms of testing, and we've been rewarded with some good work and some good luck, where some of those predictions that we made have come true, and that will allow us to, again, hopefully bring up a pretty robust set of testing for faculty, staff, and students.
Thanks, Mark. We do have some questions now. The first one is about students that perhaps have risk factors, and the question is, are there options for them to learn online?
Yes, that's been part of our plans from the very beginning, and all students have to do is let us know about that, and we'll make those accommodations. I wish I could say that every class is available online, but some classes are actually really hard to put online, but the majority of classes will be available that way. So absolutely, we did anticipate that, and it's turned out to be true.
Another question here, do you have a threshold as far as number of cases on campus that might warrant going back into complete remote learning?
We're actually discussing that literally this week, trying to figure out what the numbers would be there. I can't give you specific numbers there, and I will say that it could be that we base it on a specific number of cases. It could be that it's based on using up the quarantine space that we have for students who are going to be quarantined. But some of the scientists have weighed in and said that it should be maybe based more on the rate of change rather than specific numbers. So we're literally trying to confront that exact question this week, but I don't have a firm answer for you yet.
Will students be able to come to campus? Will they have a test prior to coming to campus, or will they just be tested when they arrive?
Both. So we're going to make that announcement. I believe it's later this week, but we're going to require students to have a negative test before they arrive on campus, and then they'll be tested several times actually once they get to campus. As Mark said, I mean, we're setting up a lab that has an amazing capacity to do testing, and it's sort of like a secret weapon for us. If we can ensure that students are not contagious, that they don't have the virus, then that puts everybody in a much better framework of peace of mind in terms of what we're facing on campus. So yes, both.
And Jim, a follow-on question to that. Once they are on campus, how often do we anticipate testing will occur?
In the short term, and again, we're going to send out a definitive announcement about this later this week, but in the short term, something like once a week for maybe the first couple of weeks. And then after that, probably more selectively, depending on what we find out, and perhaps more intensively in some areas where we have more cause for concern, and a little bit less intensively in some other areas. That's where we're at with our planning right now.
The question here about labs and wanting to know if students, particularly in biology or chemistry, will they be able to participate in labs?
I think maybe I'll take that one because it says in lab testing. You'll all have the ability you'll all have the ability to participate because you'll all be tested. But actually, great question. Yes, please. We are trying to staff up the entirety of the lab. We have hires already, but we will need, as President Dean said, it's an all hands on deck situation. There will be paying jobs for people to work in the lab, both part-time and full-time. If you're interested in that, you can connect with me by email. I am easy to find. It's mark.sadam@unh.edu. But also look at the UNH job board. We're putting jobs up there on a daily basis for this.
Thanks, Mark, for jumping in. I missed that key phrase of testing. This is a good question from a parent. So wondering if parents need to have a negative test before they come and help their student move in.
We are not requiring that. No, it would certainly be nice, but we don't really have the authority to insist on that. Again, it would be nice to do it. What we're trying to do instead is to guarantee as much social distancing as we can as students move in. So we're going to have a much more, if you will, dispersed move-in process this year than we've had in the past, so that we just don't have a large congregation of people at once, which as everyone knows, because you're all reading the same stories I am, that's one of the more dangerous situations, and we're really trying to avoid that.
Great. And a question here about how will housing and cafeterias and food, what are our thoughts on how that's going to work in the fall?
So let me take the cafeteria and food first, because we just did a dry run last week at HOCO at Holloway Commons. And depending on what condition we're in terms of the threat level, there's the folks who work in dining services have arranged to have very great social distancing as people are standing in line. There's literally decals on the floor now of each of the cafeterias, each of the dining halls, separating people by 6 feet. No one is going to be no buffets. Nobody's going to be touching food directly. People will be wearing masks. Depending on the condition, we may or may not have people actually eating in the dining halls. It may be purely takeout under difficult circumstances. As things get better, we hope that we can have spaced seating available in all three of the dining halls. Also, the menus are going to be the same in each one of the dining halls and at each one of the stations, so that there's no sort of wandering around trying to figure out what you want. It's pretty well organized. I give a lot of credit to the people who are running that. In terms of the residence halls, you cannot create social distancing among roommates. It's just not practical. And so we are treating roommates as basically members of a family. And so not trying to protect them from each other. But beyond that, we expect people will be wearing masks when they're in common areas, maintaining social distancing. If we find that someone in, let's say, a floor of a residence hall is positive, we'll do contact tracing, and that may lead to having testing for all of the people on that floor, or other similar kinds of activities.
And a follow-on question, Jim, for your statement about ham rec. So someone here is commenting on the video of the setup and wondering, is that still in place, or are buildings now being prepared for the students to return?
My understanding is that they're being now prepared for the students to return. I think we were given the go-ahead to start disassembling the hospital basically a few weeks ago. Now, I haven't been in there, but I'm told that we're taking that apart and converting it to be once again used as a recreation center. Of course, recreation centers will have to be reconfigured as well, and we have a team making some pretty elaborate plans for how we can have recreation on campus. We've just made an investment in some I'll say activities that can be used outside. As long as the New Hampshire fall lasts, we hope to have people outside and doing some activities there. But we'll also have classes. We'll do the things that Hamel Rec has done. We'll just do them in a different and safer way.
Great, thanks, Jim. A question here about international students. Will they be allowed on campus?
Yes, they will be allowed on campus. We will ask them to be tested before they return. If they can't do that, we will test them as soon as they come back and ask them to stay in isolation until then. Having said that, depending on what country they're coming from, the infection rate in the United States is actually higher than many of the countries that the students would be coming from. So they're coming from a less dangerous to a more dangerous environment. But we want to make sure that they're safe as they come in, both for themselves and for the rest of the campus community.
And maybe now a follow-up question from Mark. It's about the type of test that we're going to administer. Some nervousness about the nose test, and wondering if they could learn a little bit more about what we're planning to do.
Right. Well, we're not doing what is affectionately referred to as the brain tickle. That's the nasopharyngeal swab, the one that everybody says, I don't want to do that more than I have to. We're just doing a plain nasal swab. It's a very large Q-tip. It only goes in the very front part of the nose. So it would be nothing that would be uncomfortable for folks. And then students, everybody will be required. You'll get your own kits. You'll swab your own nose, put it in a vial, and then drop that off to the lab for sampling. If I could lead onto that one little piece, Debbie, is one of the things that our scientists have been really good at is predicting the future a little bit. And we do think that it's a reasonable guess that within the next 4 to 6 months, that even less invasive tests will be available. So you've heard about saliva tests, you've heard about antigen tests. We'll approach those when it's the right time to do it. What we're doing right now is what's called the RT-PCR test. It's the gold standard test by the CDC that they've put out. And we're gonna stick with that gold standard test until another analytical method comes up that is as good. We're not gonna back off for ease of use. We're gonna stay with the best test we can provide until something that's a little more user-friendly comes that gives that same kind of sensitivity.
And Debbie, if I can just add there, I've had that exact test twice in the last few weeks. It takes less than a minute. It's not really painful. It's just as Mark said, it's really not bad at all.
That's great. Thank you both for that. So Jim, maybe one last question. We've had a couple like this. This particular person has a very excited freshman who is eager to come to campus and wondering when we'll be releasing dates for when they can plan to move in.
Soon would be the best answer that I can give on that. That's a level of detail that they don't give to the presidents. So I'm not going to try and do that, but we will be releasing that information soon. I know the folks in those offices are making those planning. I would guess it would be within the next week or so.
Great. Why don't we end it there? And this would be a good time to turn it over to Brad and Corey.
Hello, I'm Corey Taylor. Graduated from UNH in 2011 with a degree in chemical engineering. I work at Lonza Biologics in Portsmouth, New Hampshire.
I'm Brad Miscowski, graduated UNH in 2009, also a chemical engineer, and I work for Moderna. Next slide, please. So the mRNA 1273 vaccine that Moderna is working on is an mRNA-based vaccine against COVID-19. mRNA is created based on the DNA sequence of our particular gene in a process called transcription. mRNA then contains the code cells needed to make the specific protein. Proteins perform different functions in the cells. Moderna has been working on vaccines for about 10 years. And we have, I'm sorry, not 10 years. We have 10 vaccines that are targeting different human viruses. Our approach is to take the genome sequence of a virus, pick a protein or proteins that are on the surface and make the mRNA for those. One pretty cool thing recently with the COVID-19 ramp up in production for us was we were able to take the SARS-CoV-2 genome sequence from the internet, download the sequence, and then we were able to code the spike protein, and we were able to make an mRNA and release for clinical use in 42 days, which is something that probably wouldn't be possible 10 years ago. Next slide, please. So the vaccine is injected into a patient, delivering mRNA to immune cells where copies of the spike protein are made, mimicking coronavirus infection. All proteins made by our cells have sugars added to them and fold a certain way due to the environment they are made in. Since this mimics a viral infection pathway, all the decorations and folding will be the same as when you are being exposed to a live virus. Much different from growing the virus in a mammalian cell culture, the same protein will never be made in your own cells using that. Next slide, please. So with mRNA-based vaccine, there is no viral manufacturing. The patient uses synthetic genetic material to make their own vaccine, accelerating timelines from bench to bedside. Another amazing way we prime to have a shot at pulling this off is our work with Merck on our personalized cancer vaccine. The technology we needed to invent to make a unique vaccine for each person's cancer was directly applicable to mRNA 1273 development. So it's a great example of how research can be useful for things not expected. Next slide. And this is a good segue to pass off to Corey, but Moderna and Lonza, we had an engineering partnership for the development of the vaccine is how I met Corey. Some things we're working on are procurement and qualification of new equipment, knowledge transfer from Mrna to Lonza, and then also Lonza's CMO experience back to Moderna as we are new to ramping up production at the speed and size of what we need to make for the world. So I'll pass it off to Corey now to talk about Lonza and Moderna's relationship together.
Thank you, Brad. Next slide, please. So you guys have probably all heard about Operation Warp Speed, and I really wanted to kind of talk about, the details behind that and what traditional timelines look like and what we're doing here at Lonza. So I'll kind of start with the background of a traditional vaccine. The timeline there is usually 13 years. Once in history, we've made one in 4 years, and our goal here is really to make one in 12 to 18 months in response to COVID-19. So to explain the piece of it where we're scaling up really all of the equipment, the processing, the entire process for Mrna is called technology transfer. That's typically achieved in 12 to 18 months and is also a very aggressive timeline, something we're constantly trying to refine. The objective here is to get that done in 3 months from Mrna to Lonza tech transfer, in addition to understanding their processes, getting our people trained. We also need to outfit our suites with the proper equipment. Some construction is required in those suites, making sure we have the correct utilities. And then, of course, making sure everything runs. But kind of some of the strategies we've employed, as a site at Lonza, it's the #1 priority. So if I ask for anything, from any other assets, we have 5 assets there to give a little bit more background on Lonza, what we are. We're a commercial drug manufacturing organization. So currently we make a lot of active pharmaceutical ingredients. We have 4 assets that do that, and I'm in the 5th asset, which is called cell therapy. Just now in cell therapy, we're working on mRNA. So it's a new technology to Lonza. But those other 4 assets that are within the site are also supporting us in a lot of big ways, and as are the supporting functions. So really, anytime we need anything, people are basically dropping whatever priorities they had in order to help us achieve this timeline. Not only is that happening internally at Lonza, but the same thing at Moderna, and the same thing with all of our vendors, which kind of brings me to our strategy in leveraging strengths and relationships. There's some equipment manufacturing companies in Switzerland that we're leveraging with one of our sister sites. And there's other vendors in the Cambridge area where Moderna has a strong relationship. So it's really leveraging that and also conveying the fact that everything we're trying to get is for a COVID-19 vaccine. Doing that's really enabling us to expedite the timelines for equipment procurement and a lot of other things. In addition to that, as Brad stated, we are highly focused on collaboration and standardization, benchmarking across sites, and basically in order to achieve that. And you see a peregrine falcon off to the side. That's our that's our name for the project at Lonza. It's kind of a flying bird's a symbol of hope, but we're trying to do this project faster than ever. So that's the fastest animal on earth, right? So that's kind of how we're getting a little bit of extra motivation from the team digging deep. Next slide, please. So wanted to provide some progress as to how we're doing at Portsmouth. And really so far, we have successfully designed and retrofitted 2 suites with over 100 pieces of equipment. We've, executed the automation, commissioning, and validation of the equipment. So, tuning the different equipment, getting it to operate as we intend. And then one thing Brad and I have really collaborated a lot on is establishing maintenance, calibration, and spare parts plans. Basically benchmarking what Moderna has versus what we have at Lonza that we already have existing, if there's any similar pieces of equipment, taking that with what the OEM recommends and kind of taking the best of all 3, combining that and reviewing it as a team to make sure all parties are aligned so that we can carbon copy. So it's kind of working smart and collaboratively together. In addition to that, there's been a lot of work to develop SOPs, training documents, to set up new parts. And as a result, we've actually achieved the fastest tech transfer in Lonza history. We produced our first development batch in 3 months. So it was a success. We hit that timeline. And we also set a record for the largest batch for Moderna. So it is an excellent milestone to hit, one we're very, very proud of. But there's still a lot of work to do, which takes us to the next slide. Yeah, thank you. So the next steps really are going into official GMP production. So that's clinical production. The objective is to stockpile, the mRNA vaccine so that when we are ready to actually release it commercially, we have a good head start. Another part of it is really scaling up and overseas. As I mentioned, there's benchmarking even with our Switzerland site. They're a player in this too. So it's working, with Moderna and with our Switzerland site to collaboratively help design the equipment, determine what we're going to do for factory acceptance tests, what type of controls would improve the performance of the equipment and, protect the quality of the product and consumers. A lot of it is also facility design. We're retrofitting existing suites. There's also a good bit of new construction. And all of this is kind of happening in parallel as we get, the first stage of it up and running here at Portsmouth. And then procurement, again, a lot of emphasis on that. That's probably one of the biggest, repetitive risks is, how do you fabricate highly specialized equipment in such a short period of time? We're trying to be commercial manufacturing by 2021. So there's a lot of that. And again, the next step would be that commercial piece, pending a lot of other steps, but we're hopeful we'll get there and we'll achieve that in 2021. And then after that, we can finally celebrate as things go back to normal. And, you can stand next to people at the grocery store without thinking that they're killing you or something, So that's really it in a nutshell. We'll hand it back over to Mark.
Great, thank you. We have, there are a couple questions. One was about the production numbers, but we'll get to that in just a second. We wanted to take just a short sidetrack here and talk a little bit about how UNH works with industry. As we all know, our job, universities create and disseminate knowledge. That's what we do. And some of that work is done in partnership with industry. So I wanted to walk very briefly through how we look at our industry partnerships and then talk a little bit about what we're doing with Lonza before we go back into the Q&A session. So when we think about how industry can play a role in the education or research or experiential learning of our students and faculty and staff, we look at these 10 different vectors. And these are all the ways you would imagine that industry support would be really useful. So obviously there's job placement and research and my version of technology transfer, which Corey talked about, which is the licensing of intellectual property out of the university into startups and into large businesses across these 10 different vectors. Now, there are more ways than just that to work, but these are, I would say, the most common ways that we see university industry engagement. So when I get the next slide. And what I wanted to focus on was, with my cute little green checks, is that our work with Lonza actually hits a lot of these already. And I'm going to go into just a brief bit of detail in terms of the different ways that UNH and Lonza have formed a really excellent partnership over the years. For those of you who aren't in the Seacoast region now, or don't remember where Lonza is, it's basically off of our back porch. Lonza is about 6 miles from the university. We graduate a lot of students that wind up finding jobs in Lonza. We do a lot of work with them. And we found over the years, we've made a very intentional engagement with the site to be of service, to find ways that, not beyond providing workforce, how are there different ways that we could work? And the best part of corporate engagement strategies is these are two-way streets. So can I get the next and final slide for me? I just wanted to talk a little bit about how that, the different ways that manifests itself. So the first, obviously, is we are the largest creator of workforce in the state of New Hampshire. We're also the largest importer of diversity into the state of New Hampshire. And so we have a really important role for the industry in our state. And we provide, there are a lot of UNH alums at Lonza. The good news is the number keeps going up. As a percentage, it goes down. At one point, I was told a few years ago that it was upwards of 30%. But as Lonza grows and is a major employer in the Seacoast region, that percentage goes down, but more UNHers are over there. We do some research, actually, through this program that the state funds called the New Hampshire Innovation Research Center, where industry comes to the university with problems that it needs to solve. And so we have active research collaborations with the site locally. We also provide some key services to Lonza with our analytical support. So we have instrumentation. The university has 1,500 or 1,600 pieces of high quality lab equipment that we make available to industry on a contract basis. And we serve that role for Lonza in different ways that waxes and wanes over time. But we've very consistently been providing some useful services to Lonza for the last few years. They were really critical, and I cannot thank the site enough, Corey and Mark Caswell, who's the GM at the site, of providing very strong support for the renovation, or I call it the renewal, of Spalding Hall. As a proud biochemistry alum in 1993, it needed the work that we're going to do to it. So it's good to see that Spalding will be renovated, and Lonza provided a lot of support locally and in Concord to encourage the importance of Spalding Hall to the state. And then, as you might imagine, as my wife likes to refer to it, in COVIDity, there's a lot of activities that are going on, and the government is really trying to help. There are lots of grant programs. There are lots of projects that the government would like to see accomplished. And so UNH has been very aggressively pursuing grant opportunities to not only provide economic recovery, but also resilience to the community. And we are working with Lonza on a grant with the EDA that talks about how we could do some virtual training, how we could put things in place in the state that will last far beyond the creation and the implementation of a vaccine, which we're all very grateful for the people on this call for. But I think we all recognize that work won't quite ever look like it looked in February, and that virtual learning, these kind of environments, are not necessarily always a bad thing. And so we'll be working on some large grants with the support of Lonza and other companies around the state to provide some resiliency for this kind of activity as we go forward. So I just wanted to give the folks on the 204 participants on the call a little bit of a taste of how we like to work with industry. And if you're on the call and you are part of the New Hampshire industry, if you find ways that you'd like to engage with the university, please do reach out. And then the last piece, which is not quite Lonza related, but back to the lab that we talked about earlier, is we do have some work that needs to be done in terms of how we process samples and kits. And we're actively trying to do that work with New Hampshire companies. So when we have needs to satisfy, we're trying to work with the New Hampshire business community wherever we can to satisfy those needs. So some of you might be hearing from me when I have needs to facilitate the opening and the testing of campus. So keep your ears out for that. And I think with that, we'll move to questions. And I'm gonna stick with the first one because it was great. And it says, did any of the work you did at UNH prepare for the work you're doing now? Were you able to conduct experiments while a student that helped develop the skills that you're currently using? So Brad and Corey.
I would say, yeah, absolutely. Especially, I'd say collaboration, right? Understanding being successful in college was a lot about understanding that you can't do everything and that you need to utilize your team. Whether it was a lab group or something to that extent, a lot of it is delegating in order to make yourself successful and making sure that you have all the bases covered. I'd say general engineering principles are definitely always something that we lean and rely on and almost take for granted. As you're talking about, any of these processes, we can get into heat transfer, other, fluid dynamics, that having that background really enables us to understand and quickly figure out whether or not solutions are viable. Brad, I'll let you answer as well.
Yeah, I agree with Corey. Getting through chemical engineering, a tough program at UNH, you have to lean on your classmates, understanding concepts, assignments, things like that you have to kind of understand or learn and grow together. But I would say just all of the principles, kind of like Corey said, mass balance, certain things that you do take for granted when you're learning it as a chemie. But then when you get out into the real world and you're dealing with equipment and processes, whether you're in a lab or you're working on a floor in manufacturing, it's funny sometimes how concepts and things that you've learned either from, a professor or a random thing that you wrote down as a note that maybe has applied to a boss that's kind of taught you on that concept. I mean, I would definitely say that, the engineering disciplines and the degree has definitely helped, I would say, Corey and I grow in our careers.
And as a recovering biochemist and also an alum, I was actually educated by Rick Cody, who is now one of the faculty members helping us bring the testing capacity up. So I learned everything from Rick, and it's been great. So actually, the next few questions are fairly related, and it really is about the production of the vaccine itself, and they're very straightforward, which is what is the volume that is expected to be created, and when will that be available, and do you have any idea of how the quantity to be created at the point where basically it becomes accessible to everyone, assuming positive phase 3 results?
Yeah, so that's I would say kind of at the forefront of the relationship with Moderna and Lonza, because at Moderna itself, we have our own manufacturing suites that, we have a certain amount of capability that we could put out of our building in Norwood. Our headquarters is in Cambridge, but we just built a new facility 2 years ago in Norwood where we're ramping up production now. And then, Portsmouth, and then as Corey mentioned, the sister company, being able to get worldwide production. The exact numbers right now, I'm not exactly privy to all of that, but I know I've heard numbers that we want to have, 500 million vials of the vaccine in 2021 at some point. How that comes to be, I think, like you said, Mark, we need to get through, inspections and readiness, for both sites, for both companies, to then be able to have that success and hopefully get it approved to be able to, distribute to the world.
Right. So go ahead, Corey. Sorry.
Yeah, I was going to say a lot of it depends on, what the final dosage is determined to be, right? However, whatever quantity is actually required for injection is going to kind of dictate it. But the original, goal was, as Brad said, 500 million up to 1 billion doses a year. So definitely a hefty goal, but I think we have a good plan.
Right. So the less of the vaccine that you need to elicit the appropriate response, obviously, the more vaccine you can create. So if you need a high concentration, production goes down. And remember,
Exactly.
for the folks asking the question, there will be more than one solution likely. There are at least, I think, 15. There are 150 vaccines that have been proposed. There are at least 15 that are beyond that phase 1. And just to give some context to the scale of what we're trying to do, the phase 1, I have the press release from Moderna at my left here. The phase 1 trial, which is safety, which is just to make sure things are okay, was 45 patients. And now we're talking about a leap to 1 billion samples, or 1 billion vaccines in under a year. If we're successful, it will be the single greatest accomplishment of humankind to be able to deliver a vaccine from diagnosis to delivery in that short period of time. So thank you both for the work that you're doing there.
Yeah, and I just want to
go ahead, Corey.
I want to clarify one thing. You said there's several different vaccines being produced. This is the best one. Okay.
Without question.
That's a good answer.
It's got that wildcat magic on it.
That's right.
I didn't even have to pay him for that.
We
Get your
all
back,
agree,
Brad.
trust me. The next question is, any idea how long the vaccine would be good for? I mean, I imagine that means the length of the immune response and what the goal is.
I mean, I think that's something that we're still learning right now, actually, with even the testing that we're finding out, people becoming reinfected. So that's something that's, on the front of our mind with our vaccine to understand the dynamics of that. So, I mean, ideally, I guess you would like to get vaccinated once and, basically have it be done with from there. But I think that's something that we still don't know, worldwide on how people become reinfected, how long, you can elicit the response to, be free of the vaccine. But it is something that we're still honestly determining as we are developing and ramping up production.
So the next question was, well, there was one about multiple times as the flu. Look here. Sorry, there was one question, I just skipped past it. Oh, here's a question from earlier, President Dean. So it says, sorry, going backwards, required testing students need before returning to campus. For resident students, commuting students, every day, how do they turn it in? I can answer a piece of that if you want to answer the front end of how frequently.
How frequently will students need to be tested?
Yes.
So we're still working on the exact numbers there, but as I mentioned in the early stages of the fall semester, roughly every week. But that will be adapted based on what we learn from that.
And the second part of that question was about the difference between residential students and commuter students. And I know we mean all students. All students will be tested. Faculty and staff that have direct contact with students will be tested regularly. Oh, there were two questions here about distribution. I'm not sure, Brad or Corey, that you can answer that, but there have been questions both here and nationally about, well, one question was, would UNH get preferential access to the vaccines? But I think that there's a related, please, but there's also a related question of just, a solution in Portsmouth and Cambridge. Who's going to get those? How is that going to be distributed?
So I can only really reiterate what I've read on the news about that. If, really being focused on manufacturing is where my head's been, but what I've read is a couple different things. That the, early vaccine or phase 3 trials will be used to vaccinate frontline medical personnel, right? One of our most valuable resources and highest risk groups. And another thing that I've, read a little bit about is setting up different sites in different cities that have a high likelihood of having an outbreak. And in the event that those cities do have an outbreak, those would be used as the sites. So, so the answer to that would be hopefully not UNH. Hopefully we're continuing to show a decline. But, I think it's anybody's guess. Brad, do you have anything to add to that?
Yeah, I'd say it all kind of goes back to the approval process for Moderna holistically. With government agencies, if we're able to, apply for or get an emergency use case, then they might, expedite kind of to what, Corey said, to kind of find these hotspots to be able to provide vaccine there. And obviously the medical industry would benefit from it because they're on the front lines of defense for, everybody. So it would depend on, I guess, our approval process. And then maybe if there's any spikes or anything like that, that could expedite us being able to sit with the government agency to be able to, figure out what we can do, to show whatever results we have at that moment in time.
Okay. Next question from Greg Tucker. What IP with government and academic collaborations are Moderna and Lonza licensing? So where's the origin of the technology? Has any of that originated from academia?
I'm not sure, Brad, you can take that one.
Well, we have a partnership with BARDA, a government agency, We were able to get the genome, as I said in the presentation, to be able to develop something in our preclinical manufacturing to get a clinical type of the vaccine created in 42 days. So, our relationship with BARDA is enabling us to, A, create the vaccine, and then B, be able to fund it and, produce and have partnerships with Lonza to ramp production. But the mRNA technology is Moderna, that's our platform. So we were just able to, use our vaccine history with different other products that we have, and then, be able to develop the response to COVID based off our technology and just, our resources in our library as a company.
And I can answer a little piece of that the early technology that Moderna used was licensed out of MIT, out of the Langer lab. So it does have an academic provenance, and that was federally funded. It was about 10 years ago when Moderna was started. So let's see. How is the team handling at Lonza? I will extend it to and Moderna, handling such a volatile, uncertain, complex, and ambiguous situations. Any advice for managing and leading teams through these challenging times with reference to manufacturing?
I would say there's a lot of people working really long hours. And one of the things the teams who are working so hard continue to reiterate is when they look across the room, they see other people doing it too. So I think one of the biggest things for us is feeding off of each other's energy and understanding we share, the same goal, and also the impact this has on so many lives, right? I'd say there's never been quite as much pressure on, our teams as there is now to deliver something. And it's something we take extremely seriously. So as far as, coaching people up to kind of get to that point, I'd say a lot of positive reinforcement and just thank you. But I'd honestly say we can't do enough to say thank you to the teams who are working this hard to achieve it. There's people, putting in very, very long days and weeks. And but I think everybody gets it. It's not really difficult to motivate the team on the Lonza side of it. We're all highly motivated.
Yeah, I mean, it's motivating for me every day, just getting out of bed to work with Lonza and the people within Moderna to back on what Corey said. I mean, we're trying to create a vaccine to get some normalcy back in the world today, right? So if you can't get up for that, then I don't really know what else there is to motivate you out there, And from Moderna's perspective, we're hiring people, so if people are looking, fresh students out of school or whatever it may be to help with that manufacturing, we are hiring, and and especially from our site, we've been kind of ahead of the curve with the social distancing and things like that. We've been wearing masks for a while on site. We have a temperature screening on site every day, so we've taken the necessary precautions even before the CDC put everything out there in the individual states and things like that. So not only are we taking it serious, but we're also protecting the people that have to show up every day, because you can't make production behind a video screen. You have to be there working on the equipment. So just like UNH is, we're protecting the people that are making it, and obviously just trying to motivate them as much as we can.
Great. I'm going to combine two questions here and just say, so how does this compare with other vaccines that you've tried to scale up, and have you seen, or do you think that have you heard of any response that's different between age or ethnicity? That one might be for the clinicians, but I figured I would ask because it came up a few times.
Yeah, I don't have a great answer for that one.
And this is the first vaccine I believe we've produced at Lonza. We're typically not a vaccine company.
Yeah.
So it's unique.
Moderna has 10 vaccine candidates where we have, I think our Zika virus is one of the ones where we've kind of progressed the most through, but that again is with help from the BARDA government agency, so we've been able to, work with them. But all our other vaccines are kind of under the normal timelines for approvals and things like that.
Yeah. There are a few, another couple of questions that I'm going to shrink. President Dean, this is a little bit back to you. So turnaround time for results of COVID testing outside of the UNH lab to be is running 7 to 10 days. So is there a process for what's going to happen if people take the test but can't get the results back in time? And then related to that, what is the process that will follow if students or groups of students are identified as being COVID positive, and how will that be managed both academically and physically?
Right, so both great questions. On the first one, we're going to ask people to time the test, so there'll be a very high probability that they'll have the results back before they return to campus. If they, for whatever reason, and we can certainly understand, there may be reasons why they don't have the test back, we would ask them to isolate until they do, so that they're not putting their classmates and other people on campus at risk. And similarly, I think if I understand the second question is what happens if you get a positive test. So we have put aside hundreds of dorm rooms, residence hall rooms, for students who need to be quarantined. They'll be able to take their classes there to the extent the classes are online. They'll receive medical help, probably mostly through telemedicine. They'll have meals delivered. We've been working on that for months. So we will have a very good situation. Now, of course, if students decide that when they test positive that they would prefer to go home and being taken care of there, and if they and their parents are on the same page with that, then obviously that would be fine too. And it's one thing, you know, if you live in, I don't know, Concord, you can drive home. But some of our students live in California, and we can't, in good conscience, allow students to get on a plane. And so they really would have to be housed in our quarantine facilities.
I'm gonna ask and answer one that I can answer. Actually, a few times people said, "What's the turnaround time if UNH's lab goes into operation as we expect? What's the turnaround time?" And I'll say that we feel pretty confident at this point that we'll be able to get results within 48 hours, if not sooner. And our objective is to get that down much sooner than that. But obviously, we're not done building this, but definitely 48 hours, ideally 24 hours. And then possibly sooner than that. It's part of that strategy. So, great question. Trying to understand better how this vaccine is different from others. Can you explain?
Brad, do you want to take that one? I think the simple answer is it utilizes the mRNA platform, right? Rather than a barely living or dead virus, right? So the implicit risk of actually having the symptoms is reduced, and it makes it more scalable. Brad, I'm sure you can get a lot more technical on it than I can, though.
I mean, you covered it pretty good. Yeah, the mRNA platform, again, you know, it's eliciting the response with a protein from that mRNA coding, and then once it's injected, it finds the spike protein as part of the COVID-19 virus, and then reacts accordingly. Again, that's very simple to say, but there's also, you know, managing your immune response to that as well, to, you know, fighting the virus, but then, you know, not eliciting too much of an immune response, you know, because it's a natural response when you're using the mRNA.
As opposed to what I would call a traditional virus, which is usually a either fully dead or an attenuated virus, to quote the Princess Bride, mostly dead, but that's how those work. As someone who went to Jonas Salk Middle School, we learned all about the pileoid virus. So, all right, we have, are there any mRNA vaccines currently in use right now?
We don't have any commercialized products, Moderna yet.
And there aren't any, and I know there are not any other mRNA vaccines on the market yet. Another question, can you talk a little bit about the race to be first? To get a first company for, or even the first country to complete a vaccine? What do people say about the pressure?
Yeah, I mean, internally, I guess internally it's hard dealing with all the kind of unknowns that come on a day-to-day basis. as Corey and I, talked about in our presentation, it's just the speed that everything, that we have to go at. We're in a compliant industry though, right? We can't cut corners. There's no ways to, make it faster and better, but you're, not following the rules, because that's not what happens in this industry. So, the speed and unknowns to me the hardest things to kind of deal with on a day-to-day basis. But again, we have, smart people within the company to be able to kind of adapt and learn from experience that we've had, either working on the Zika virus and the ramp with that, or just as a company trying to get one of our products commercialized. This has helped us, put our name on the map even more than it already was. But it really, again, I think, like I said earlier, it's hard not to get motivated for what's going on and seeing, the economy in the world today with, being affected so much by this virus. So, it's tough, but we'll get through it.
The latest question was, we're hearing a lot about the trials that happened on Monday. Can you tell us how that went? It sounds good.
Yeah, they just released additional phase 1 data. So I haven't read completely into all of that, but, all of the data looks really good, and we're able to, basically be ready for our phase 3 this month for, I think, 30,000 patients is what it's supposed to be. So we're, getting ready for that. And then, starting with, Corey's group to be able to start the first equipment train there to start making as much virus as we can, and then ramp up to the other, sister company within Lonza.
Great. Oh, this is a good one. As a mom and alum with a child in engineering, very excited to see your professional progress. What kinds of things did you do on campus while here?
I participated a lot in sports, like did the intramural sports. I played hockey my whole life, so I always loved going to the hockey games and things like that. Met a lot of really good friends on campus. I'm actually from Massachusetts originally, but decided to go to UNH. So I met a lot of really good friends that I still have today from, just being on campus every day. And when I first took a tour with my father back in the day, I really fell in love with just kind of the small campus. But even though it's kind of a big school, with a lot of people that went there, a lot of, really nice towns and areas around. So, it's just a beautiful area to be.
Yeah, for me, it was a little bit different. I did a few years of electrical work before going to college, and then I went to NHTI before transferring over to UNH. So a lot of my time I spent working in parallel with going to classes, but really had to work less, especially junior and senior year. So a lot of long days on campus. We did have our own intramural soccer and hockey team, the Chameleons, which is about as nerdy as it gets, but really developed some great relationships with some of the people there. A few of them are actually on this call right now, so shout out to you guys. Thank you for attending. But yeah, I'd say it's hard not to make good friends, especially within the Chemi group. You spend so much time together working on projects. So I have no doubt your child will have a good experience at UNH, make lots of friends.
I think the biochemistry class of '93 basketball team would give you a run for your money on the nerd names, but I'll just leave that. So let's see. Sorry, one of the questions got. Oh, it was how did Lonza and Moderna find each other? How did that connection happen?
That's kind of funny. I'm not sure exactly. I wasn't privy to all of the conversations, but I was lucky enough to be one of the first people on the Lonza site to basically meet the Moderna team, was able to give them tours of our facility. And I'd like to think I helped kind of get them on the hook. But I think a lot of those decisions also happened at, executive committee level, type of conversations. I think a lot of it had to do with the commitments that Lonza made to deliver to these timelines. I think was probably one of the biggest drivers. And I know Moderna had also made commitments to BARDA in order to achieve certain timelines. So I think probably the biggest thing was enabling them to actually do it. And so far, so good.
Yeah, to
Okay.
the point, I wasn't necessarily involved with, how it came to be with Lonza and that decision, but it's just kind of funny. I actually introduced myself in one of these engineering teams that Corey and I met. And, next thing I'm here on a webinar with you guys. So it's kind of cool.
Okay, we have one last question that we'll do. It says, is there any concern that there may be long-term side effects from the vaccine that have not gone through lengthy safety testing that other vaccines have been required to do?
Well, we're not skipping any, phases or anything like that to, a normal vaccine production. It's just we're doing it with a little bit more speed. So as you said earlier, Mark, phase one is purely safety. That's what phase one is for, to test all those things, ensure that it's safe. And then as you go through the other trials, phase 2 and 3, you expand the cohorts and who you're giving it to see those immune responses. But that's what phase 1 is the test safety. So yes, we are going faster, but we got data that showed that what we based our phase 1 on, it was safe. So there shouldn't be any concerns with, just because we're doing it faster, that it's not as safe.
And I think it's a bit of a risk-based approach too, right? The government is ultimately, at the end of the day, the FDA in particular, is going to determine how quickly we're able to go through phases and go to commercial, right? So there is going to be a review of it, but you thinking about that from my kind of a common sense perspective, right? What is the risk of implementing the vaccine versus not doing it, right? The alternative is to continue to fight through this pandemic. So, my personal speculation, not representative of the company I work for, is that that's going to weigh into some of the decision making at the FDA level. But to Brad's point, yes, we are not taking shortcuts as a company, as a team, at Moderna or at Lonza.
For what it's worth, the phase 1 trial was 45 people. That is not an unusual size for a phase 1 clinical trial. That is a perfectly normal size for a phase 1 of any kind of therapeutic. So everything is going according to plan. I think as data science gets a little better, what they're doing is a phase 2/3, which is a combined study, because data science is able, phase 2 is efficacy and phase 3 is sort of large populations. And they're able to do that much quicker because it's easier to pull data quicker and sort through it quicker than we used to do it. So with that, I'm going to hand it back to President Dean to close.
Thanks, Mark. Thanks so much for that. So since I'm addressing Mark, I wanted to thank Mark publicly, both for facilitating this session and then also for the tremendous work that he's done in getting these laboratories stood up. And it's just unbelievably elaborate and hard work over a long period of time. And really the health and safety of our entire campus community is going to benefit because of the work that Mark has done. But of course, Mark has a day job as well, and you've seen the results of it here, which is our connection with companies around New Hampshire, starting new ones, connecting to existing ones, and so on. So Mark, we're in your debt for a lot of really good work that you're doing.
Thank you. It's a big team. It's a huge team that's doing this all over campus, but thank you.
Yes, and that was a very wildcat response. It's not me, it's the team. So I appreciate that. Similarly to Debbie and her team, on the call we have Susan and Jen and Phoebe and lots and lots of people have been connecting us with our alumni, trying to make sure that those connections remain and that people know what's going on at the university. And that's really important. It's good to see. It would be easy to sort of back off and say, we'll get back to you when the crisis is over. But instead, you all have doubled down in terms of the connections with alumni. And it's certainly something to be proud of. And I'm really proud of you for doing that. And then in particular to Corey and Brad, thank you so much for joining us today. It's great to hear about what you all are doing. And you're a bit poster children, not you're grown-ups, I get that, but for UNH alums and for people that have taken their schoolwork very seriously, gotten your degrees, gone out and not only having productive work, but doing something that really is making a difference in the world. And we're really very, very proud of both of you. And thank you for sparing time today to work with us. And then to all our alumni, thank you for taking part in the conversation today. We had at the high point, I think a couple hundred people joining us. Really happy to have you connected with us. We hope to continue to do these. We don't want to be isolated. We want to try and reach out as much as we can. And we thank you for devoting the time to learn about your alma mater and learn more about these projects. So with that, thanks everyone. Be safe, wear a mask. Bye-bye.
Thank you.
Bye. Thank you.